everyone, it's Mr. Richmond. I'm working on this week's audio video reviews. I guess it's just audio this time because I don't have my webcam working. I can't seem to get it to work. But either way, this is our mitosis and meiosis review for biology. Term 4, continuing on going over items that we've spoken about before. We will be covering today cell cycle and mitosis. We're also going to go over mitosis versus meiosis, and then at the end go over different examples of each, asexual and sexual reproduction. So, cell cycle, mitosis, and cytokinesis. The cell cycle itself is a characteristic of life. All living things have to grow and reproduce. Cells go through this process called the cell cycle, and we have three main parts to it. Our three parts are interphase, mitosis, and then cytokinesis. Interphase is our longest stage. This is just when cells are being cells. Mitosis is the division of the nucleus only, and cytokinesis is the division of the cytoplasm. Pretty much everything else. Now, if we look at interphase, we can break it down into three main parts. We have a G1, an S, and a G2 phase. So, we come over here. Actually, let's change this over to pink. So, we have M for mitosis. So, let's go ahead and block this off. And if we notice, we can see all of this right here is interphase. All of this. Interphase is by far the longest section of the cell cycle. We have our G1 phase, which is going to be a growth phase. So our cells are going to grow. That's why we call it G1. The synthesis phase is where we're going to replicate more DNA. And then G2, we're going to prep up for mitosis or for division. We got to get a couple more things, maybe duplicate our centrioles or maybe make some more mitochondria. It's getting ready for the division. Notice it is a much smaller phase. So, if we look at our chromosomes right here, the ones that we coiled up, we have a few things that we have to keep in mind. We have an entire side of the chromosome. It's two strands of DNA connected together at a centromere. Each one of these strands of DNA is a chromatid. That's going to come back up in a moment. But S phase, normally they're just a chromatid just hanging out by itself as a chromosome. When we go through S phase, we duplicate it, but we don't want to lose it or we don't want them to get mismatched, so we keep them connected together here at our centromere. So we still have 46 chromosomes, they're just now duplicated. Mitosis, again, we said it's the division of the nucleus only. After DNA replication and interphase, the cell has double the number of chromosomes. Now we have twice as many as we had, a bunch of copies. And this occurs in four main phases. So it occurs for our first phase, prophase. Let me go ahead and write these out right here. Chromatin turns into chromosomes. It coils up. The nuclear membrane disappears and spindle fibers begin to form. Metaphase, we have our spindle fibers attached to the centromere, the center of the chromosome where that X is connected. The chromosome, or the sister chromatids, they move the chromosomes into the middle. So a good way to remember this is metaphase, middle. Then we have anaphase, where the spindle fibers pull the chromosomes apart. Another way to remember this. So anaphase, apart. And then we have telophase. Chromosomes are at opposite ends of the cell. The two nuclei begin to form. What happened here in prophase begins to undo in telophase. So the chromosomes are going to uncoil back into chromatin. The nuclear membrane reforms. The spindle fibers disappear. A good way to remember this, PMAT, is Patty made a taco. Quick, simple little alliteration. So here is our cell cycle all strung out. We have our interphase, prophase, metaphase, anaphase, telophase. If we look at these four, this is our mitosis. If you've noticed, we put a lot of effort into learning about mitosis. It's really, really, really important, especially for the DNA. Remember, the DNA is usually held nice and safe inside the nucleus. Now it's out and exposed. We've got to be very, very careful with it. And finally, cytokinesis. Telophase shows our final division of the nucleus, whereas cytokinesis, we're splitting into two different cells. So, not all cells separate the same. Plant cells do not have centrioles. They do have spindles, but they don't have centrioles. Animal cells divide using a cleavage furrow. So one of the things I like to talk about with this right here is a cleaver. So a cleaver is a large knife that butchers use to separate big pieces of meat. And if we think about it, animal cells undergo division using a cleavage furrow. They cleave or they separate into two cells. It pretty much is like a ball or a underinflated balloon that you pinch into the middle and eventually forms two. Our plant cell, though, we can't just pinch it in the middle. An analogy I like to use is the plant cell is like my classroom. So I can't just push this wall over here out of the way into the next classroom to make two rooms out of my room. I would have to actually build a new wall right in the middle to make two different rooms. And that's what we do here with a cell plate. That's how we divide our cytoplasm in plant cells. Mitosis versus meiosis. So, getting into our differences between the two, now they're both going to be making new cells. However, the process of meiosis and mitosis produce very, very, very different types of cells. So, meiosis occurs in gametes, or what we know as sex cells. Mitosis is going to occur in somatic cells. Gametes are only found in one area of the body. However, somatic cells are all of the other cells in the body. So we want to have a lot of these. There's many, 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 many different types. As far as humans go with gametes, we only have two different ones depending on the gender. Meiosis. We have haploid cells. They have half the set of chromosomes. We know this is formula N. So there's a reason that we want them to have half. If we take two pieces, two parts, an egg and a sperm, are two different gametes, and combine them together, if we had a full set of chromosomes in each one, we'd come out with way too many. If we had 46 in one cell and 46 in another, we'd have 92 total, which is not something that we want. So haploid, it's important to get these down to half their original number, 23 and 23. In humans, our normal set of chromosomes is 46. We get that out of mitosis. And if we go back and look at it, we have a diploid set, or a 2N set. And if we look at our formula for N, N actually equals 23. So, in meiosis, our gametes are going to divide two times. We're going to have PMAT, PMAT, prophase, metaphase, anaphase, telophase, two times. The nucleus in mitosis, however, is only going to go through it once. Because we went through it twice, we come out with 4 daughter cells. In mitosis, we only come out with 2. And here's our biggest, most important distinction between these two. Meiosis. Our daughter cells are different than the parent cell, and they are different from each other. We want to make sure that all of our cells are different. We want genetic differences, variability. We don't want to clone our genes from one generation to the next, because if we do and we have a genetic disorder, we can't get rid of it. In mitosis, though, in body cells, we don't want a bunch of different types of cells. We want the same cell from one to the next generation. So our daughter cells here are actually going to be identical to the parent. All right, and if we look at this graphically, we have our mitosis here on the left. We have our mitosis here on the left. You can tell because it's only got one division. Our daughter cells come out genetically identical to the very original. And on our right over here, we have meiosis. We went through our division phase twice. We need to have 4 cells at the end. One last graphic representation. Notice we have 4 chromosomes up here that have duplicated, and after our split, we have 4 in each one. Our original cell over here with meiosis has 4 chromosomes. After 2 splits, each one only has 2. Meiosis occurs only in gametes and in 2 stages. We have meiosis 1 and meiosis 2. They actually do have a very short interface between these two, but it's extremely short, and we don't get rid of everything on the way. So sometimes we'll see it in textbooks, and sometimes we won't. There are 2 major things happening during prophase 1 and of meiosis 1. And our first and biggest things is that homologous chromosomes pair up. So instead of getting a straight line of chromosomes like we did before, we're actually going to have chromosomes sitting side by side. Homologous chromosomes pair up and they form this thing called a tetrad. This prefix tetra means 4, so we have 1, 2, 3, 4. 1, 2, 3, 4. Let's change that color. It's not going to work. There we go. We have 1, 2, 3, 4 strands of DNA. So we have 4 pieces of DNA in this one pairing. That's why we call it a tetrad. And this process that happens is called synapsis. This crossing over is going to occur. If we look down here on the bottom right, you can see that we are actually going to swap pieces of the chromosomes. They're going to transfer over. This is even more genetic variability. The name of the game with meiosis is we want things to be different. We don't want 2 cells to be the exact same. We want as much difference as possible. All right, and here is our depiction of meiosis, our long form of meiosis. And if you notice, we have prophase, metaphase, anaphase, telophase 1, and then PMAT 2. So let's get over here to prophase 1. If you notice, it's very, very, very similar to what happened in, oops, let's undo that, what happened in our mitosis. Chromosomes coil up, the nucleus disappears, spindle fibers form, and centrioles move. Now, this is also where our synapsis is going to occur. We're going to form those tetrads. So that's one major difference. And if we look over here at metaphase 1, you notice we have these 2 colors right here. And what that is just those homologous chromosomes lining up. Instead of being one long straight line going down the middle, it's 2 pairs. It's a pair of chromosomes lining up the middle. So then we look over here at anaphase 1, and we notice that it's pulling the entire chromosome apart. So if we look at it, instead of it breaking at the centromere, it's actually pulling an entire chromosome, still connected to the centromere, across to the ends. And then telophase 1, just like normal, we split the cell, cytokinesis actually. We uncoil the chromosomes a little bit. We don't do super amounts, but the main thing here is that we've now made 2 cells. So then we look over here at prophase 2. Notice we have 2 cells. And actually, if we look at the ends of our chromosomes, we did have crossing over occur previously. So prophase 2. Now, we have half the number of chromosomes, but we still have these intact centromeres, so we need to split those this time. So again, if the nucleus had formed, we dispense with that, we create more spindle fibers, and our centrioles move again to opposite sides. So metaphase 2, just like normal, we would line them up in the middle. Anaphase 2, we split them at the centriole. And telophase 2 in cytokinesis, we undo everything that we did during our prophase. Make a nucleus, uncoil chromosomes, and get rid of our spindle fibers. Now, if you look at it, we have 1, 2, 3, and 4 cells created out of this one original parent cell. Spermatogenesis versus oogenesis. So obviously, not everybody does both of these. These are going to be male gametes, and these are going to be female gametes. The big difference between spermatogenesis is we get 4 viable sperm cells, 4 that will be useful and necessary for reproduction. Each one of these can get used. However, oogenesis, we only make 1 egg, 1 good cell, and we have these 3 polar bodies. These are just kind of leftovers, things that we don't need. This is quite literally an example of putting all of our eggs in one basket. So, spermatocytes divide through meiosis 1 and meiosis 2. We get these spermatids that will then mature into becoming sperm cells. If we look at our oocytes, notice our first division here, we have 1 really, really large cell and 1 very small cell. We want as much in this bigger cell as possible because it gives us our best chance of survival at the end. And again, we do another set of meiosis. We unevenly divide a second time, so our cell is as big as possible. These are very, very big. All right, so asexual reproduction. This is very similar to mitotic divisions. Not all of them are the exact same, but they are all going to be asexual. They don't need 2 sets or 2 different cells to do this. We only have 1 parent. Our offspring are genetically identical in the sense they are clones, and we do not use gametes here. There's a lot of good reasons to do this. It's very quick, it's very simple, and if you are an organism that doesn't need that, like a bacterium or a yeast, you can just split right in 2. Doesn't matter. So, binary fission. Again, this is bacteria splitting into 2 equal-sized offspring. They have the same number of DNA. All you'd have to do would be to copy your DNA and then split into 2. We have budding. This is what yeast do. This is going to be splitting, resulting in 2 unequally sized offsprings. However, that smaller one is still going to have everything it needs to be a functional organism. Spore formation. This is going to be when plants and fungi are in environments that are too harsh to survive in, so they are going to produce a bunch of spores that they can then release off into the environment to make sure that they can survive. We have this vegetative propagation. You can think of grasses or strawberry plants for these. And then regeneration or fragmentation. The big example we'll use for this one is going to be worms or starfish. So here's our binary fission. We take our DNA, duplicate it, and split our cell right down the middle. Can't get simpler than this. Our budding, this is a hydra. This is an aquatic animal. We have a small bud forming off. We have a new hydra, and this detached hydra is, in a sense, a very tiny version of the original. The same thing happens here in yeast. We duplicate our DNA, split our cells, and we send our offspring off to be its own thing. All right, asexual reproduction. Ooh, no, let's go to the next one. As far as spore formation is concerned, this is the fungi spore formation. The sporangium here is going to be a pod that produces a lot of spores or specialized cells that are going to be identical to the parent that can get released in the environment. These, if it gets too dry or it's too hot or cold or whatever happens, the fungus can produce spores so that it can make more copies later on. Vegetative propagation, when you take a piece of a plant and it's able to grow or make an extension from either a root section or an old clipping of a plant. And regeneration and fragmentation. So this example right here is fragmentation. this planarian right here. So I know it says regeneration right here, but honestly, this is fragmentation. We split it into multiple pieces, and each one of those pieces grows a new organism. Even this little bitty tail section over here will grow an identical copy of the original organism. Now over here, with the starfish, notice it's lost a limb. It's going to regrow. Actually, we can see it growing a little bit right here. This is regeneration. So we've lost an entire limb. Now we need to grow it back. That's going to be regenerative. It's going to grow it back. For some reason, I cannot say that word this morning. All right, sexual reproduction. Meiosis is going to be forming gametes in this. We have to have two parents. We can't do this without two parents. Offspring are going to be genetically different from the parents and each other. We are going to be using gametes. We have 4 examples of this. Fertilization. Fertilization is a general overview idea. It's the union of male and female gametes during sexual reproduction. We have a pollination that occurs. This is going to be mostly in plants, where the sperm in the pollen is fertilizing the eggs that would then result in seeds. We have conjugation, which is when bacteria exchange DNA. And then we have this idea called alternation of generations. Plants can get pretty confusing sometimes. They have some have an entire stage where they grow an entire plant that is haploid. So we'll get into that in a moment. So fertilization. We have an egg and a sperm. They are both haploid. They come together to form a diploid cell known as a zygote. We have gametes and zygotes. Pollination. So we have a male portion of a flower, the stamen. It's going to produce pollen, and a pollen is just a granule with a sperm cell on the inside. A pollinator, whether it be a bee, a fly, or even the wind, is going to take this piece of pollen and bring it over to the pistil of the female flower, the female part of a flower. It's going to land right here on the top, and it's going to allow the sperm to come down here to the eggs. Conjugation, the transfer of DNA from bacteria. So this is actually one way that we can get bacteria to become antibiotic resistant. You've heard of the superbugs, the ones that are resistant to all the drugs. Well, this is how they pretty much share their information. They'll create this pilus. It's almost like a walkway between the two bacteria. They will copy and send a section of a small piece of DNA known as a plasmid over, and it's a good analogy would be actually a DNA playing card. He's trading cards with this guy over here. He's giving him a copy. So they both have a copy of this plasmid, and whatever information was on it, they now both have. And alternation of generations. We have a mitotic stage and a gametic stage of plants. Some plants go through this, where they will have a multicellular diploid stage. We are diploid. Humans, most of the cells in our body are diploid. Now, instead of just producing one cell that then goes over and fertilizes, like we saw in pollination, we have an entire plant that grows up haploid. So we'll have one plant that is 2N that produces a plant that is just N that then combines with another plant and produces another 2N plant. So alternation of generations. Again, plants being a little bit stranger than we're used to seeing, but still it's a good way to trade up. So that is it for a review of the mitosis versus meiosis and looking at different forms of reproduction. Again, Mr. Richmond, and have a good day.